I just had. Okay, well, I would say our Saturday field trip was a success.
Oh, yeah.
It came out as good as they ever do. They had an intern that they finally scheduled for us, and he was real enthusiastic and showed us a lot of different things in the collection. And then, they had redone the way they present stuff at the vertebrate zoology, and I thought that was the way they did it now was better. You know, they had examples of cool things in the museum laid out when you went back into the collection. And that, I think that worked out even better, so that was good. There was a really nice exhibit there about Jane Goodall. That was very interesting. I learned a lot. You said you read a book on it and you thought you learned more from the exhibit?
Actually, yeah. Because, yeah, the book was more for like studies, but this was more like her life history.
Yeah. Huh. But anyway, that was fun. And then we did I didn't get to spend much time. I took almost all my free time in the Jane Goodall exhibit. But this was that. And we did get to see the Deinosuchus. I estimated the length on that, and my rough estimate, just looking at it, was 35 feet long. A lot of that's the tail. But that was fun. Did you guys want to share anything about what you enjoyed on the trip? No? Okay.
Go first.
Go for it.
I like they had brine shrimp there. I never seen like brine shrimp before.
Oh, is that right?
Yeah.
Where was that, up on the upper floor?
Yeah, that was right by the paleo section.
Oh, it was by the paleo.
Skeletons.
Oh, okay.
The triumphs.
Well,
The triumphs.
triumphs? No, the brine shrimp. They were like little crustaceans near, there was like these other exhibits where there were like tubes of water, sculptures of living animals. It's actually right, where you're looking at that angle, it's right before you walk
Over here?
towards the crocodiles, yeah. Or a little bit farther back, so you're at the brine shrimp and then walk straight through the
Mhm.
walkway.
Oh, yeah, there's that the thing on Great Salt Lake, was that the exhibit and the
Yeah.
brine shrimp wrap? Yeah, I had a graduate student who was a herpetologist for the state of Idaho. And then there was what do they call it, the Cowboy Caucus, where the right wing pretty much took over and they fired like 30 of the biologists down in Utah. And they reassigned him to studying brine shrimp. And because that's actually a commercial thing there, you know.
Brine shrimp?
Yeah, the shrimp, yeah. And so he couldn't take, he wanted to study herps, so he came up here and worked on his PhD. Okay, well, anyway, that was good. Our next field trip, I'm gonna try to do it. Boy, I think a week from Saturday, I think. Cause I wanna get it in before you guys take off for spring break. And 3 weeks from now, you're gonna be, I think, in spring break.
Yeah.
So we're gonna try to get that. How many people, what would that be? That would be, anyway, not this Saturday, but the following Saturday. How many people are interested in going to the Living Planet Aquarium?
That would be amazing.
One, okay, raise your hands up high.
Will that be the 9th, March?
Yeah, it would be the 9th. Cause the 16th would be right before the break,
Yeah.
and sometimes people like to take off early. And at least that's been I don't know so much in herpetology class, but like, I remember at Thanksgiving when we didn't have the whole week, people would leave like on Wednesday.
And based on the tentative schedule you made, the 12th is scheduled to be a lab exam as well. So the field trip Saturday and a lab exam on Tuesday, is
Uh-huh.
that right?
Yeah, well, see, when we get back from spring break, you said the ninth? Yeah, right?
The ninth.
Yeah.
Yeah.
When we get back from spring break, then you're gonna be really working on your projects. And we're gonna be having weekend field trips to go to the field. So I wanna get all of I wanna have all those weekends after spring break open for us to go into the field if the weather is nice. And sometimes the weather's crappy and we never get in the field at all, which is really the worst thing.
Yeah.
Oh, because that's even better than going to museums and things is going out and seeing live stuff. So. Anyone else wanted to say anything about the field trip? I will have photographs of that and I'll post a PDF on the website. So let's see, today we're going to work on water relations. We hopefully will get that done. Tomorrow we'll have lizards of the world, and then you'll key out US lizards. There's the dragons, yeah,
So you said we had the lab exam on the 12th, which is during spring break.
I think
No, it's
spring
not.
break is the following.
The
Yeah, it's the
following.
18th.
Yeah, it's before spring break.
Okay.
And then there's if we have any extra time tomorrow, we'll have the Dragons of the Dry video from the Life in Cold Blood series.
Like Komodo.
Oh, yeah, he's got really cool stuff with monitors in there. He's got one, it might be in the very first one that we haven't seen, but I'll put that up as an optional one. And he's got this interaction with this monitor in the field in Australia. And it's a big monitor. And it comes up and it gets on its hind legs and it just sits there and looks at it. And he's talking to it and pointing at it and it's looking at it. It's really a neat clip. We'll do Tuatara on Wednesday because it's a Lepidoptera, so it goes there with the lizards. And then on Friday evening, you're going to have to have submitted your title and like a 1 or 2, or however long you want to make it, but at least a 1 or 2 sentence description of what your student project is going to be. And I'd like to go around the class on Friday and have you just tell the other people in the class what you're doing. That, keep it to a minute. So, and then we'll start gas exchange. Oh, and then I should have the evolution exam done by tomorrow night. So I'll post that. And then you'll have a week to work on that. So any questions about what we're doing? Lots to do. Okay. So I don't know, got maybe a third of the way through on water relations. On Friday, we talked about why water is important. And then we cast it in this idea of, just like, remember how we talked about thermal budgets, that we could write an equation and said, you have the temperature gains and the heat gains and the heat losses, and that equals storage. And then you can actually solve that equation. You can measure all the things that are affecting that. You can do the same thing for water. It's harder, but you can do the same thing with water. So I think I'll just start there again. So the way that you gain, or animals gain water, is through liquid water, which they can drink or they can absorb. Preformed water is water that's in their food. And metabolic water is water that's produced by your metabolism. When you digest food, it actually, like when you take a sugar, break it down into carbon dioxide and water, right? That releases water. So that's actually can be an important source of water. And then the losses are things like evaporation, water that's lost in getting rid of nitrogenous waste and urine, small amounts of water with feces, and then secretions from glands. And then the hydration state or the storage is, you know, where the animal is when you add up the gains and subtract the losses. Okay, so we talked about drinking. And that most amphibians don't drink, but most species of reptiles do drink. We talked about lizards drinking droplets of dew. We talked about animals that got water from condensation on fog, this Namib viper, about rain harvesting behavior and viperids. And then how some turtles can use their shells to collect rain and in the skin of these mollusk lizards so that they can direct water that falls on the skin to their mouth. And then I think this was about where we ended up, I think. Is that right, Jessica? So, you know, amphibians, most amphibians, they're sitting there like a free water surface. They're almost like a saturated sponge or something. They're losing a lot of water. Western toad can lose half of its body water in less than 2 hours if it's out in the open on a hot day. And they balance that by bringing in water across their ventral surface. So most terrestrial anurans have what's called a pelvic patch, and it's an area of high permeability to water here in this pelvic region. This is a Western toadlet, and this dark area here, most of the water that they absorb is going to come right through that part of the skin. Okay, arid land amphibians can absorb water faster, so here's one of your spadefoots. They're gonna be really good at picking up water. Let's see, this is for the western spadefoot, Speya hammondii, spend most of their time in the burrow. And then what they do, this is a really cool thing. I don't know, some of you have had comparative physiology, right? Is that right? No, okay, well then I better explain this. Some animals, like sharks, do this. You know, your problem when you know, for most, you know, for like most fish or mammals would have the same problem too, is that the ocean water, for example, is saltier than your body fluids. And so what's going to happen water-wise? In the water, are you going to have trouble losing water or over gaining water, picking up too much water if you're, say, a bony fish? What's your osmoregulation problem?
Will it be that you, intake more water? Because if the, sea has a higher concentration of salt, so high concentration will go to a lower concentration? Or not?
The water is more highly concentrated in the body of the fish.
Oh, so I will.
So the water, their problem is eliminating salts and conserving water. Okay. And sharks, the way sharks are chondrichthyan, you know, cartilaginous fish do it, is they let urea, which is a nitrogenous waste product, they let that build up in their body fluids. And then that prevents the water from being pulled into the from leaving and going into the sea. It's basically why you can't drink salt water, right? You'll die of thirst in an ocean, if you're in a raft or something like that. And it doesn't make sense. You think, well, I'll just drink the seawater, but you'll actually end up making things worse by doing that. Well, spadefoots have a problem that, they're going in this burrow and then they may be in there for 9 months and they're going to be, you would think they'd be losing water to the soil. But what they do is they raise the osmolality of their body fluids by doing just what the sharks do in the ocean. They let their urea levels, nitrogenous waste levels build up. And so as the soils dry out, they're building up their level of urea and they still can, conserve water or even absorb water from the soil, which is pretty neat trick. So this actually shows the bladder with dilute urine would be what, 100 goes to 300 millimoles per kilogram of water, but the blood plasma that can go up to like 600. And so that prevents water from being pulled out of the toad by, or the spadefoot by the dry soil. Cutaneous uptake, let's see, this is under hormonal control. There's a arginine vasitocin, AVT, that stimulates water uptake across the skin, and then it suppresses urine formation. So this is affected by hormones. In freshwater, the problem is just the opposite of what we were talking about in the ocean. You end up taking up too much water, so they have to produce a large amount of dilute urine and actively absorb salts and ions via the skin and the gills. In brackish water, you'd have the opposite problem. Oh, someone was asking about this. There's 13 species of salamanders and 61 species of anurans. I didn't remember it was that many, but can occur in brackish water. And they use the same trick too. Decrease water loss by increasing their osmolality by accumulating solutes such as urea. Okay, with reptiles, their skin is far less permeable to water. It's thicker, it's got keratin. For reptiles, it to a large extent, they're more terrestrial and they don't have as much water loss across the skin. So they're not really, most of them can't uptake water very much. The skin lipids form the main barrier to water movement. So there are, lipids in the skin. And I had a friend that studied water loss in birds. And he told me that he was showing me some plots of water loss as a function of temperature in these birds he was studying. And when it hits about like in the low 40s, I think Celsius, all of a sudden you just get this huge spike of water loss. It's not a linear relationship. And what it is it breaks down those lipids and then you don't have that barrier to the water loss. And I imagine a similar thing. I've never read it in the literature, but I imagine a similar thing probably occurs in reptiles as well. Only some adults are able to pick up water through their skin. We had some Malayan pit vipers, Enkystrodon rhodostoma was what they were called at the time when I was a research assistant. And they're a viper, but they're not live bearing. They lay eggs, but they coil around and they protect the eggs. They have parental care. And I was just weighing the females and the eggs to see, what they were absorbing. And the eggs can, absorb quite a bit of water. But these females actually picked up weight too. The mothers picked up weight too. So this I know from personal experience that they can absorb across their skin. And then I've read that speckled caimans can do that. Spectacled caimans. And as I just mentioned, water absorption is very important for reptile eggs. They may double their mass or their volume, after they're laid. And then food is, particularly, like for desert reptiles can be a really important, form of water or source of water. As we looked at last time, like 60 to 80% of the prey is water. In plants, the water content's more variable. And there was a very interesting study that Warren Porter and his colleagues did, and they were using those temperature thermal budget models to predict the activity periods of desert iguanas. And what they found was that in the springtime, it worked really well in predicting when they'd be active. But then in the late summer, even though their temperature models said that they could be active, they found that the lizards weren't active. So they weren't regulating their body temperatures at preferred levels, even though they could. And what they finally, I don't know if they actually tested this idea, but their interpretation was that the plant material, what they were feeding on, the vegetation in the desert, dries out later in the year. And so they weren't getting, it was costing them more in terms of their water budgets to eat the food, because they had to get rid of the salts, the excess salts. So they just quit eating. And that, at least that was their interpretation of what was going on. I think if the food had high water content, they would have continued to feed. And this kicks in here, this idea that they have the salt glands, that if they pick up too many salts, extra salts, then they have to use their glands to eliminate them. But that requires some water. I mean, they can get rid of a lot, but it still requires some water to secrete those salts. Okay, and then the last source of water gain is metabolic water. So that's formed when hydrogen from chemicals in the food combines with oxygen. So if you take a gram of starch, that gives off a little over a half a gram of water when it's metabolized. A gram of fat will give off over 1 gram of water, and a gram of protein, less than a half a gram of water. And that variation depends on what waste product is being, nitrogenous waste product is the animal's using. Anyway, that can be an important source of water. Now, the flip side of this is water loss. And so as we mentioned, that's evaporation, urine, feces, and glandular secretions. And you have what's called sensible water loss, which is like the urine, and I assume that's from the gut. And then insensible, you have skin and respiratory tract. Okay, so there's actually quite a bit. Now, an interesting thing, because of the low metabolic rates of ectotherms, what do you think the implications are of that for water loss in reptiles, say compared to mammals or birds?
Less occurring through the gut.
Well, the gut and through the lungs, right? Because they require far less oxygen, so there's a lot less air that's being moved in and out of the lungs, probably like 1/10 the amount. And so they're not going to have as big a problem with water loss off the respiratory surface. Okay. Okay, so this is kind of an interesting way to think about this. This is an analogy where they measure resistance to water loss in terms of seconds per centimeter are the actual units. And do you think of it as a resistance here? It's sort of an analogy to an electrical circuit, and this is like a resistor. And the value, the amount of that resistance varies by species. So Sormelis obesus, you'll key one of these out tomorrow. That's a chuckwalla. It's a desert animal, like from the Mojave Desert. It has very, very high resistance. Like what is this, 1,500 seconds per centimeter? So, um, let's see, I'm not sure what all of these things are. These are snakes. This actually believe it or not, is a frog. It's one of the waterproof frogs from down in South America. This is an Anolis lizard. Here's the alligator. Now, this is an aquatic animal, so its resistance is not nearly as high as like a desert animal. And then like here's a soft shell turtle. Here's hylid frogs like our Pseudacris. And then down here, even though we tend to think of like toads as being more terrestrial or spadefoots, they actually have really low resistance. So if they're out of the water, they're really losing. And I think I told you that one of my students did some water loss experiments on uh, Western toads. And I think he calculated that within 2 hours, they could lose half of their body water. So they had to be really careful. And that's what his research showed. He looked at different logging practices on the Targhee National Forest, like, clear cuts versus thinning versus old growth. And they can't spend much time out in the open like that. And then he put transmitters on them, tracked them around, and then measured water loss rates with physical models. And one of the telemeter toads traveled a couple of kilometers away from the breeding pond, and he was heading along and hit a clear cut, went into the woods, went around it, and then came back out the other side and then continued on. So that can really affect, where they go and when they're active and how long they're active. Anyway, in amphibians, cutaneous water loss is pretty high because their skins are so permeable. And the aquatic amphibians have thinner skins and have higher loss rates. Some species may decrease by secreting mucus. So if they can put a watery mucus there, that can slow down. The bullfrogs have been known to do that. They've measured that. So some species may decrease by secreting mucus, and some may actually form cocoons. So this is what the Australian burrowing frog, African bullfrogs will do that. And I'm not familiar with this one, Lepidopatrachus. But I know that one of our ceratopheres, I had one as a pet, and it went into an estivation, and it was just like a cellophane wrapper around it. And then when it came out, boy, it was brilliantly colored. It was really cool. These are wonderful animals here. These are waterproof frogs. Let's see, I don't know. I wonder if this will work. Let's see. Now it's spreading that wax all over its body. Anyway, that shows you. Yeah, not bad. So there's a African racaphorid that does this, and then these phylomedusid frogs from down in South America. There's some other very interesting things they do. They have a thermal preference that's like a reptile, like 37 degrees. Their thermal preference is like 10 degrees Celsius higher than most amphibians. And they use uric acid as their waste product. And then they can just turn almost completely white to reflect sunlight. So they have a variety of things. Some animals like coqui frogs there are known to change their posture. So if they're trying to conserve water, when they're resting, they will bunch up and decrease their surface area. Reptiles, generally, some of them, like we were talking about here, like this chuckwalla, have very, very high resistances. I think desert tortoises would be up there too. But they're still variable, and that's correlated with the habitat. One of my graduate students looked at water loss rates in snakes, and he found that rubber boas have the highest water loss rate. They burrow in the soil, and they're in pretty wet litter usually, or moist litter. And then garter snakes were kind of intermediate. Gopher snakes had very, very low water loss rates. So I think this is from your textbook, 100 times variability in resistance to water loss going from tropical to desert in legless reptiles. A student that I knew, Steve Waldschmidt, he was studying Euta stansburiana, side blotch lizards, which you'll be keying out. And we might even get to see some in the field if we go over to the birds of prey area. That they have 25% of their water loss occurs through their eyes when their eyes are open. So he said when they were trying to conserve water, a lot of times you'd see them in the field and they'd be basking and things, but they'd have their eyes closed. The flip side of that, did I mention about using the eyes as brain coolers in
As what, coolers?
pilot that cool the brain? I don't think I mentioned that. Yeah, that was a really neat study that Shoemaker did. He I think it was with a Hyland frog, and they put tiny thermocouples like in the brain, and then they used an infrared thermometer to measure the temperature on the eyes and on the skin. And they watched as it heated up, and when the brain temperature got to a certain threshold, then they opened their eyes. And they can drop the brain temperature by 2 degrees Celsius by evaporative cooling from the eyes. Okay, respiratory water loss, lower in ectothermic herps because they have lower metabolic rates than endotherms. And then if these animals go into estivation and decrease their metabolism, their body temperature further, then they can really low. And it turns out, I thought that water loss in burrows was going to be a big problem for reptiles, but when I was working with Warren Porter, he told me that once you get, even in the desert, if you get down in the burrows very far, they're almost 100% humidity in the burrow. And so that there really isn't that much water loss, evaporative water loss there. So here's the next one. This is a real interesting story. And this is an interesting story just in vertebrate biology overall. And that's the loss of water by having to create some form of urine to get rid of nitrogenous waste. Okay, so when proteins are broken down, you produce nitrogenous waste when the amino groups are removed from proteins during digestion and metabolism. And the simplest of these nitrogenous wastes is ammonia. The problem with ammonia is it's toxic, and so you can't let it build up or it'll kill the cells. Urea is kind of intermediate, and then uric acid is a solid and it precipitates out, and it's relatively inert. So we'll go through, let's see here. We'll go through, let's see, hold on just a sec. I'm missing some of my notes. Whoop. There we go. So we'll go through those, but anyway, this is this coloring here, this shading and these dotted lines indicate which ones use which, or what we think they do. So these real aquatic animals here, like osteichthyon fishes and agnathans, they use ammonia because they have a lot of water available and they can, they can easily dilute it and get rid of it. But the reptiles, and let's see, many of the reptiles and the birds here and stuff, they use uric acid. And then the ones in gray here, they use urea, and that includes amphibians and things. Okay, so here's a comparison by nitrogenous waste product. So ammonia is water soluble and it's energetically very inexpensive to synthesize. So if you can get rid of it by diluting it down, that's the least expensive way to do it. So that's what a lot of the aquatic, the nitrogenous waste product that a lot of aquatic animals use. But it's very toxic, so you got to get rid of it fast. Used by larval and aquatic adult amphibians. Bullfrog tadpoles, about 90% ammonia for their nitrogenous waste product. And remember the South African clawed frog, Xenopus levis, they, even the adults are using a lot of ammonia as a waste product because they're highly aquatic. Okay, urea, and here's the chemical formula for urea, is also water soluble, but that requires ATP to convert ammonia to urea. So it's more energetically expensive, but it's less toxic, so it doesn't require as much water to get rid of it. So that's the main waste product for adult terrestrial amphibians. So in those bullfrog adults, 84% of the nitrogenous wastes are gotten rid of through urea. And turtles and crocs use urea when they have access to water. And then the last nitrogenous waste product is uric acid. And that's inside, when a bird poops on your car, that white stuff is not poop. That's uric acid crystals. They use uric acid as their nitrogenous waste product. Relatively insoluble, it precipitates out as water is reabsorbed in the bladder or the cloaca. And then it's excreted as a semi-solid. So any of you that have kept, pet lizards or pet snakes, you've had to clean up the uric acid. Non-toxic, little water required for excretion. Remember most of the amphibians are using, the adult amphibians or terrestrial ones are using urea. But the Phylomedusid frog actually uses it. That allows it to conserve water even more. And then turtles and crocs use uric acid when water is scarce. And then squamates primarily use uric acid. Reptile eggs also use uric acid, so that uric acid builds up here in the allantois. Okay, that's the waste, where the waste products, that's where that the uric acid builds up, and it's inert, it's non-toxic, so, and it doesn't require, much water to get rid of. So, and it basically just accumulates in the egg shell or in the egg. Okay, the last form of water loss then are the salt glands. And let's see, you're seeing salt glands in a number of organisms. This is a desert iguana here. So it's trying to conserve water because it's, it is living in the desert. This is a sea turtle, this is some type of sea snake, and this is the saltwater crocodile. And so this red here is showing you where those glands are located that are getting rid of the excess salts. Okay, salt's coming in from food and from water, and it secretes it. It's not diluting it and getting rid of it in the urine. It's secreting it through these glands, and almost dry secretions, so it doesn't require a lot of water. So this occurs in a lot of marine reptiles, so like sea turtles, crocodilians, sea snakes, and marine iguanas do this too. When you see marine iguanas, they'll often have salt built up all around their nostrils. Desert lizards and green iguanas have this too, because they get so much salt from the plant material they're eating. Let's see if this link works. They're blowing the salt that builds up in their nostrils, sneezes. I think I told you when we were down in the Galapagos, we were walking along the rocky shorelines, and you just see this, them just this. Okay. Well, the links are treating us well today. Okay, habitat selection, particularly going down into burrows, can have a big influence on water loss rates. And that's probably why a lot of the desert animals spend so much time in burrows, is to reduce water loss. Okay, water may be stored as urine in the urinary bladder of some amphibians and reptiles. So there's the urinary bladder there. Okay. In anurans, they may be very large. They may accumulate up to 60% of their overall body weight in their bladder in terms of water. I don't know, have any of you ever had that experience where you like pick up a toad and it pees all over you?
Yeah.
Yeah, that's something that they do. They use it as a defensive mechanism, but then the problem is they just lost a lot of their water, and they may or may not be able to make it up easily. That size of the bladder is related to how terrestrial and how arid, terrestriality and how arid it is. Some animals resorb water from the bladder to maintain normal water levels of other tissues. So they actually are storing that water and they can reabsorb water from the bladder. And that's, the coqui frog is an amphibian that's known to do that, and the desert tortoise. My understanding too is one of the reasons why you don't want to mess around with desert tortoises, if you like find one out in the desert, is that they may use that as a defensive strategy too, and they'll dump their water, and then they might dehydrate and die later in the year because of that. So if you see desert tortoises, you should leave them alone. Amphibians have lymph sacs. I don't know how familiar you guys are with lymph vessels, but you know, in normal circulation, or what you normally study is you know, you have blood pumping from the heart through the arteries and then out into the capillary beds, and fluid is forced out by pressure, and primarily by pressure into the interstitial fluids. And then most of that water comes back into the into capillaries and then into veins, and you know, so you have a continuing circulation of that fluid. But you have a buildup, there's some of the fluid that doesn't get reabsorbed. And so there's a blind-ended parallel circulation system called the lymphatic system that's bringing that excess interstitial fluid, which is just like plasma. It's bringing it back and dumping it into the veins so that you can, you know, recover that fluid. Where apparently lymph sacs are important for removing excess water that accumulates in the tissues in amphibians. And this diagram here shows locations of where those lymph sacs are located. And then apparently in chuck walrus, they can actually use their lymphatic system to store water. Last thing I think here is dehydration tolerances. Osmotic balance is adjustable. In us, you know, we really not only regulate our temperature really well, but humans regulate their pH very finely, and we regulate our osmotic, you know, levels. Those can vary tremendously in some of the amphibians and reptiles, very broadly compared to amphibians and reptiles. Here's an old paper, but just showing you dehydration tolerances in a variety of amphibians. So there's a spadefoot. They have the highest dehydration tolerance of any amphibian that was measured in this study. Western toads were close behind. Remember, these guys have real high permeabilities, but they can tolerate a lot of water loss. And then intermediate here, and then these are the aquatic ones. Okay, well, I've run over time-wise here. I'll finish this up on Tuesday. It'll only take about 5 minutes or less to finish that. Okay?
I heard of the